Hi, my name is Angelo Chan, and in this video series, I will explain how next-gen sequencing data is commonly processed. The slides in this video can be downloaded in the links in the description below. Let us begin. I'll begin by giving some background information on the topic before explaining what the core pipeline is. I'll go over the pipeline in an increasing level of detail, how to get to the pipeline from alternate starting points, how to set up the pipeline, and then some additional technical background information. In the context of biology, sequencing is the process of figuring out the DNA sequence of a strand of DNA. Sequencing has been a thing since the early 1970s, but in the early 2000s, a series of new sequencing technologies became commercially available, which had exponentially more throughput than previous technologies. These are known as next-gen sequencing, or NGS. Now, there are specific scenarios in which some of the older sequencing technologies perform better than NGS, but at least for now, we're not going to cover that here. The main point I want to make is that, generally speaking, all NGS data undergoes the same initial processing procedure. This is what I refer to when I talk about the core pipeline, and this is what I'll be going over in this video series. There are cases where this core pipeline doesn't quite work, such as when the DNA has been modified in some way, but even then, the procedure for processing that kind of data is usually just a modified variant of the core pipeline. Hopefully, by understanding what the data looks like at each stage of the core pipeline, you'll be able to understand why it doesn't work for certain kinds of data, and how the pipeline would need to be tweaked for it to work. I will start by giving an overview of the pipeline before breaking it down in more detail. And then, as indicated in the table of contents, some parts of the pipeline will be expanded upon in a second level of detail. I organized the information this way for two reasons. Firstly, because I didn't want to bog you down with minutiae until I'd given you the big picture. That way, you can better understand how these small details fit into the bigger picture. And secondly, because the second level of detail is only essential for someone who wants to perform the analysis themselves. If you are working with a bioinformatician who does the actual analysis for you, and you just want a better idea of what they do, then the first level of detail will be enough to get you going. That being the case, you would still benefit from learning how it all works. Apart from the core pipeline, I'll also go over the setup process separately, since the setup only needs to be carried out once per genome assembly, whereas the core pipeline needs to be carried out from start to finish for every dataset analyzed. Now, for anyone hoping to do some next-gen sequencing experiments, this video series is primarily bioinformatics. I will be covering the biochemistry behind some of the wet lab processes, but only on a need-to-know basis in order to explain the rationale behind certain parts of the pipeline. If you want a detailed guide to the relevant wet lab protocols, such as how to prepare a biological sample for sequencing, this video series is not what you need. As the name suggests, I won't be covering the many different downstream analyses which can be performed once the data has gone through the core pipeline, since the workflow can diverge significantly. I am also going to assume that the organism from which the NGS data was obtained is one with an official downloadable genome assembly. If no genome assembly is available, you'll need to assemble one using de novo assembly before the core pipeline can be carried out, and I won't be discussing the de novo assembly process here. The last thing to mention before we begin is that at detail level 2, I'll discuss caveats and examples which are specific to the software programs I use or am familiar with. However, those steps in the pipeline don't necessarily have to be carried out using the programs I mentioned. Alternate options exist for every step of the core pipeline, so don't be surprised if you read papers which mention an entirely different suite of programs which haven't been mentioned in this video series. I also won't include any example code in this video series, since the goal is to explain how the core pipeline works on a conceptual level, and not to give explicit step-by-step instructions on how to execute it. Such an example, along with the beginner's guide to command line, can be found in separate files. Let's start with a brief overview of the whole pipeline. We start off with the original FASTQ files, which contain completely unprocessed sequencing data. The FASTQ files undergo pre-processing, producing data which is still in FASTQ format, but is more reliable after having undergone trimming and filtering. These sequencing reads are then aligned against the reference genome, producing a SAM file, which contains raw, unprocessed alignments. These alignments then undergo standard post-alignment processing to produce sorted, filtered, aligned reads, which are now ready for analyses which are more experiment-specific. Now, I know that, technically speaking, we might start with something that's even less processed than a raw FASTQ file. In that scenario, you'll have to derive a FASTQ file from that raw data. And that will be covered after we finish going over the core pipeline, so don't worry.